Hello, good afternoon, viewers at home. Today, January 4th, 2025, is another opportunity once again to be able to meet that we ought to know in biology, irrespective of our field or area of specialization in biology. Are we molecular biologists, microbiologists, environmental biologists, wherever, at any time and anywhere, a geneticist? We This is a very important topic, and this time around we are going to look at it from the molecular aspect of it. That is, what really happen at a molecular level, so that everybody, every one of us, anytime we come across such terms and terminology, we will be able to actually have a very correct perspective or glimpse about it. So just like I said in one of my earlier video, that I actually made mention of the genetic causes, or what you call genetic disorder. And one very important thing, and that is what we want to consider today, and that has to do with molecular basis molecular basis of point mutation. So we want to consider mutation, and we want to focus on point mutation as well. So um, once again, this is another opportunity. We are trying to look at what is a mutation. So, and if you talk about mutation, every one of us at this point in time will know exactly what mutation is all about. So, and this time around, we want to focus on point mutations. And in trying to look at point mutation, we want to look at the molecular perspective, the molecular uh, angle to explaining point mutation. So don't forget, just as we already have it on the board, and from what we discussed earlier on, point mutation is a sudden change. When you say mutation it is a sudden change in genetic makeup. So, and when we say genetic makeup, that means it must have its basis from the DNA. So, and if it has its basis from DNA, that means it is going to it has the possibility of being passed from parents to the offspring. It has the possibility of being passed from parent to the offspring, and that is exactly what we want to talk about. But before we even get to that level, today we want to look at different let's okay, now the first thing is, what is going to be our objective? Why are we so interested? And what's going to be the scope of our discussion here? The first objective, that at the end of this lecture, we should be able to uh, familiarize ourselves. We should be able to familiarize ourselves into the basic concepts of point mutation, and its molecular mechanism, or sorry, which is a molecular perspectives. So we should be able to familiarize ourselves with the basic concept of point mutation and its molecular basis. Okay. Then number two, we should be able to recognize. You should be able to recognize different types, different types point mutation, different types of point mutations and their molecular basis. So we should be able to understand that. Then another thing is that we ought to know at the end of the day to be able, so we should be able to actually we should be able to mention, know the clinical stroke genetical implications of point mutation. So, and we also need to know the roles of point mutation, or the role of mutation generally, the role of point mutation in cancer. So that is going to be what our objective of this topic, what it is going to be like. So in order not to waste our time, let's start from understanding at this level. As a student of biology, we ought to know that DNA is a double-stranded molecule. It's double-stranded molecule, and it has different bases, nucleotide bases. So the first one, the bases are guanine, which is represented by G, cytosine, which is represented by C, adenine, which is actually represented by A, and the last one is thymine, which stands for T. However, in base pairing rule, according to Chajaf base pairing rule, C, we have a triple bond. It will be triply bonded with guanine always. So A, we have double bond with thymine. This is exactly what is called the Chajaf pairing rule, the Chajaf rule. Okay. Now, apart from that, we also need to know, we also need to be familiar at this point in time. We need to be familiar with some, what is called, we have anti-parallelism. So you need to know. We are not going to explain all this because it's what you're supposed to know. Then we also need to talk about what is called complementarity rule. Is one strand to 3' end. Then the second strand will be from 5' end. That is complementarity, is that when you have the knowledge of the basis of one strand, you'll be able to predict using the Chajaf or base pairing rule. You'll be able to predict what's going to be in the other. So apart from that, now let's first of all start with what a point mutation is. We said a mutation is a sudden change in the genetic makeup. So, and there are different types of mutation. There are different. Is going to be our focus today. Point mutation. So now, what do we mean by point mutation? That means there is a change in just a single nucleotide or a single nucleotide in the DNA. There are different types. So under that point mutation, we have silent mutation, we have missense mutation, missense, and the missense one is nonsense mutation. Okay, so under that there are other types. So there are other types of mutation, but we are going to see all this as we proceed. So we are going to basically concern ourselves with point mutation today. Point mutation today. So if time permits us, we will look at which has to do with two, which is insertion slash deletion. Okay, what the short form is called index. That is insertion and deletion. So then we will now look at the third one. We will look at is going to be duplication or translocation. We are going to talk about duplication, and we will look at the last one, which has to do with inversion. Okay, which is number 4, inversion and translocation.